So, no need to introduce myself. What I will do, though, is show you our new Centre for Cancer Immunology. So, we're opening this in September, currently recruiting. We've got any smart young faculty. We'll be making appointments to the new institute in the next year or so. Okay. Is that better? Good. So, you probably know that for the past 30 years I've been interested in this interaction. We've only been able to see it for the past 20 years or so. This is a beautiful image of the antigen presenting function of MHC class 1. Here presenting a this is another sad consequence of age. I can't see. There you go. An MHC class 1 molecule presenting a short antigenic peptide to T cell receptor. And this is the function of MHC class 1 that we're all used to thinking about. But of course, the MHC class 1 molecule at the cell surface here represents just the end point of a long and complex journey of newly synthesized MHC class 1 molecules. And there's actually, I think it makes a lot of sense to think about a second function of MHC class 1, which is its peptide selector function here in the endoplasmic reticulum. So this is the intersection of two intracellular pathways, the biogenesis of MHC class 1 molecules. I'm not going to go into detail, but it's synthesized into the ER, assembles with beta-2 microglobulin in a chaperone-dependent way, and becomes part of a large protein complex around a transporter that is transporting peptides that are generated in the cytosol, probably primarily through the action of proteasome, into the lumen of the endoplasmic reticulum. And this is a sort of a co-assembly process between peptide and newly synthesized glycoprotein, MHC class 1. So there's a peptide selector function here that's actually distinct from its peptide presenter function, and that's a sort of a paradigm that I really want to establish quite early. And we showed 20 years ago that you can dissociate these two functions using site-directed mutagenesis. So you can introduce mutations into the class 1 heavy chain that prevent this function from happening, although you can quite easily assemble those molecules in vitro and they'll present antigen perfectly normally. Okay, so why do we want to study this at the molecular level? Well, we think that it's a process that underpins many immunological phenomena. I mean, that's stating the bleeding obvious. But one immunological phenomenon is immunodominance, and this is illustrated beautifully by this this model, which is one of the immunological models that we use. It's the recognition of this tumor, transplantable syngeneic tumor CD26, by T cells. And we've mapped the response, the specificity of T cells that infiltrate this tumor. It's a rapidly growing tumor. It's not very immunogenic in a sense that it's not controlled immunologically, although there's a very strong immune T cell response to it. I don't have time to go into the details of immune regulation of this response. What I do want to do is draw your attention to the fact that about 60 to 70% of the T cells that infiltrate this tumor at day 25 recognize a single peptide. We call it GSW11. It's an 11-mer peptide with this sequence. About 20 to 25% recognize a second peptide from the same protein antigen, and this is the one that Drew Pardol identified many years ago. And there are about 5 or 6 other responses that we can map in the remaining 10 to 15% of that response. So we're beginning to understand how this can come about. So just to remind you that this peptide selector function of MHC class 1 occurs when MHC is part of a large multi-protein complex comprising the ER chaperones calreticulin and ER57, the TAP transporter, which is not in this this model here, but it sort of lies in the membrane underneath this complex. We're looking down at the peptide MHC complex here. And this really important dedicated class 1 chaperone called tapacin, one of 2 molecules whose sole function appears to be to assist MHC class 1 molecules in loading with peptide cargo. So it turns out that different MHC class 1 alleles depend to different extents on the chaperone function of tapacin. This is illustrated beautifully in this experiment done in Malini Raghavan's lab a couple of years ago, building on some work that we published about 15 years ago. So this shows the ability of a whole range of different human HLA-B alleles to assemble with peptides and present at the cell surface in the presence or absence of tapacin. Some alleles like B5701 are exquisitely dependent on tapacin for presentation at the cell surface. So this is when you take tapacin away, you see very little presentation. When tapacin's back in, you get a 6-fold increase in expression. At the other end of the spectrum, some alleles like B3503 are relatively insensitive to the presence of tapacin for their ability to load with peptide cargo and present at the cell surface. So we showed this first using 2 HLA-B alleles, subtypes of HLA-B44, B44, O2 and O5. O2 is very dependent on tapacin for presentation. O5 is relatively independent of tapacin. And we showed it using a cellular biochemistry approach. And I'm going to go through the sort of the technical platform, the sort of the assay that we've used, because it turns out that it's a very, very informative way of investigating peptide loading, even though it's a relatively, on the surface, a relatively crude way of measuring MHC biogenesis. So using an old-fashioned technique called pulse chase analysis, you can label a cohort of newly synthesized MHC class 1 molecules with a radioactive amino acid, and you can chase that over time, over an hour or so. And you can measure various parameters. There are 2 key parameters that you can measure using this technique. You can measure the rate at which MHC molecules traffic from inside the cell through the cis Golgi to the surface. And you do that by measuring its ability, the processing of the glycans that it acquires and processes as it transits the cis Golgi network. As the glycans mature, they acquire resistance to an endoglycosidase called endo-H. And so if you compare some molecular size in the presence or absence of endo-H, you can see a gradual resistance to that. And that's a surrogate monitor of transport rate. You could also measure the acquisition of thermostability. So this is the relative affinity of the total peptide cargo that's been loaded onto MHC class 1 molecules. And so the surrogate we use there is thermostability. So soon after they're synthesized, they're loaded up with peptides, but they are of a average low affinity. So they kind of melt at relatively low temperatures. You can see here we're just incubating our immunoprecipitates of class 1 at different temperatures, and they disappear at about 40 degrees. But as they mature, they acquire thermostability. So by the time they reach the cell surface and are fully endo-H resistant, these guys are pretty stable. You can refer then back to standards by doing a much more detailed thermotitration and relate that thermostability to actual peptide affinities. So I've just got 3 examples here. This is a high, medium, and low affinity peptide, and you can see that the thermostability of class 1 complexes folded with that peptide alone. And you can see that the peptide, which has a slow off rate, it's an off rate of 240 minutes with a half-time of 240 minutes, has that thermostability profile, whereas the fast off-rate peptide with a half-life of just 18 minutes has that thermostability profile. So it's semi-quantitative, this technique. At the cell surface, we can also measure the rate of turnover, so we can measure that process, which is a function of peptide dissociation. And we can measure that in a flow cytometric assay that I don't have time to go into. So the point I want to make is that you can actually parameterize MHC class 1 maturation, assembly and maturation, in quite some detail. And when you do that for a whole range of different MHC class 1 molecules, we've done about 30. I'm going to show data for 3 here, just to illustrate what I mean. At one end of the spectrum, you've got these TAPACIN-dependent alleles that don't mature to the cell surface, don't acquire thermostability in the absence of TAPACIN, but when you put TAPACIN back in the system, they function perfectly well. Other alleles that seem to be able to do this relatively well on their own without TAPACIN there, B4405, which remember is at the other end of the spectrum. And the reason I include these two is because they differ by a single amino acid at residue 116. So that's the only difference between these two molecules. They have they bind the same repertoire. It's highly overlapping, but so it's not entirely dependent. But 90% of the peptides are peptide selectivity is shared between these two. I'm also showing this allele, which is a mutation that we've made in the B4405 molecule. We did a saturation mutagenesis analysis, and I picked this one out because it reverses that phenotype. So now the B4405 molecule is no longer able to load with peptides in the absence of TAPACIN, just like B4402. So we've taken the B4405 molecule back to the other end of the spectrum with a single amino acid change at residue 147. So we were optimistic that we would see, when the crystal structures were solved, some kind of structural correlate of this difference in function. Sadly, we didn't, so we didn't solve these structures. These are provided by Jamie Rossjohn, who published them in 2003. He published the structures of B4402 and B4405 with the same peptide in the peptide binding groove, and the structures are identical, with the exception of 1 or 2 hydrogen bonds around the amino acid difference. The structures are identical. So we hit a bit of a brick wall in terms of understanding the structure-function link here. Until we met Andrew Phillips, Neil Douchow at Microsoft Research in Cambridge through my structural biology collaborator, Jörn Werner. Jörn had just come back from doing a sabbatical at Microsoft. We were testing a new language, a stochastic pi calculus language, on complex biological feedback switches. And when they looked at our sort of semi-quantitative pulse chase data, they said this is a system that we could model and try and get to the bottom of why some alleles behave differently to others. So, what's the allele-specific parameter here? Given the way that we could parameterize most of these reactions, they felt that there was a way in to investigate the allele-specific differences. Now, I'm not going to talk about that. That leads on to another lecture altogether. But I am going to talk a little bit more about this model. The bottom line is that it is possible to fit this very simple model that only has 3 components. It's only got class 1 TAPACIN and peptide in it. The key is that it's a dynamic model, so you're continually generating MHC molecules and they're continually leaving the system from the cell surface. And there's a continual supply of peptide. There's a rate of generation and degradation of each one of these complexes. It's not a closed system, it's a dynamic model. And it's possible to fit these models, there's a suite of models, to our biological data. And the bottom line is, the only point I want to make from here, is that it is possible to describe the relative abundance of a peptide MHC complex at the cell surface in terms of peptide supply, a filtration function defined by TAPACIN, the rate of egress from the ER, and the rate of disappearance from the cell surface. So that's all, that sounds obvious, but the important thing is that they're now linked in a formal framework. There's a mathematical relationship between these parameters that we can then use to explore. So we've used this model in various ways. The way that I'm going to talk about in the rest of my time is it's used to predict the relative abundance of peptide MHC complexes at the cell surface, given some knowledge of the relative abundance of competing peptides inside the endoplasmic reticulum. That's what I'm going to talk about. I can't sort of pass on without mentioning, though, that we've also used it to investigate mechanism of peptide selection. As I say, this is a completely separate lecture. But the bottom line is that the best models fitting the data include a two-state model of MHC class 1, a sort of an open and a closed, to use shorthand. So we've spent a lot of time and effort investigating the multiple conformational states of MHC class 1. And that's a good advert for coming to see me give another talk at a different time about this. Today I'm going to talk about a mechanism-based model for predicting epitope presentation. Okay, so we've heard a lot already about the holy grail of epitope prediction, which is to take a transcriptome and provide some kind of statistical likelihood of peptides generated from a new transcript appearing at the cell surface in complex with MHC and being recognized by a T-cell receptor in some kind of functional way. That's what we all want to do. At the moment, the predictive algorithms are not great. We rely almost entirely on predicting peptide affinities as a way of predicting relative cell surface abundance. And this is a study done a couple of years ago that show that while there is some correlation between predicted MHC affinity and abundance at the cell surface, it's by no means a very good predictor. So although it's fair to say that all the most abundant cell surface peptides tend to be in the top decile, top 2 deciles, there are many other abundant proteins that fall, way out of the window that we would normally capture so-called high affinity peptides in our predictive algorithms. And that means that there are other processes at play. The abundance, processing factors, transport across the ER membrane, editing into ER by ERAT1, and so on and so on. So there are two examples that I want to really focus on that just to illustrate why we think that it's important to have mechanistic models for epitope prediction that run alongside these correlative models that are derived from machine learning, which are incredibly powerful. So the first is a set of experiments that so I'm summarizing here about 5 years' work that relate the immunodominance of epitopes this is a DNA vaccine model, back to their affinity, their sort of kinetic stability, their off rate. And you can see that for this panel of just 4 peptides, which span a spectrum of stabilities from the very slow off-rate peptide through to a very fast off-rate peptide, and they're all related, but we've fiddled around with the sequence to affect peptide binding stability. We can measure the relative abundance of those peptides at the cell surface presented by H2KB, the presentation molecule. And what we find is that the relative abundance at the cell surface correlates pretty nicely with the kinetic stability of those peptides. And when we use those as DNA vaccines in a mouse tumor model, we see that the relative intensity of the CTL response to each of these peptides also follows a nice dominance hierarchy that maps back onto affinity. If, however, you take away the ER chaperone tapacin, that relationship breaks down. So now the relative abundance of peptide presentation no longer represents kinetic stability. So peptide affinity is not a good predictor of cell surface abundance in the absence of tapacin. And when you look at the relative immunodominance of responses to these peptides in a mouse model, so this is a knockout model, a tapacin knockout mouse, you see, again, this immunodominance hierarchy breaks down. It maps quite nicely onto the relative abundance at the peptide at the cell surface, but it does not relate back to kinetic stability. Okay, so that's one example. The other example is our tumour model that I mentioned right at the very beginning. So it turns out that the immunodominant epitope here that attracts 70% of the tumour-infiltrated lymphocytes is a very, very fast off-rate peptide. It's an 11-mer. It dissociates with a half-life of about 15 to 20 minutes at 37 degrees. And that's very different to what we'd normally score as an immunodominant peptide. This is one from a viral system using the same MHC class I molecule. And these are the kind of peptides that we seek when we do our peptide binding algorithms. So peptides with a half-life of over 150 minutes. So we want to get to the bottom of that. How can such a weak peptide drive such an immunodominant response? Okay, so this is the system that we've set up. It's a highly contrived but highly quantifiable system to measure the quantitation of antigen presentation from competing peptides inside the endoplasmic reticulum at different intracellular abundances. So the idea is, with some knowledge of that process, we can predict the relative presentation of these peptides. So how can we use this sort of information? Well, it'll help us explain phenomena like this. So how such a fast off-rate peptide can become immunodominant. So when you look at the genome of the CT26 tumour, you can map its mutanome. There are about 3,000 point mutations in the tumour that, when you put it through the filter funnel, yields about 150 peptides that are predicted to bind to MHC in the top 1%. But you can also look at the relative abundance of cancer testis antigens. So these are a different kind of tumour antigen. And what we find here is that by far the most abundant cancer testis antigen is a GP70 protein, from which our two immunodominant epitopes are derived. So we think that this is the mechanism for the immunodominance of these two peptides. You can see that the relative abundance of this protein to other cancer testis antigens is very, very high. It's close to the most abundant protein inside CT26. Why is that important? Well, just as sort of almost as an aside, it turns out that T cells that recognise this particular epitope are therapeutically the most efficacious. So we've tried various ways of eliciting T cells to this epitope. What I haven't told you so far is that although this specificity is the most abundant in tumours, they're switched off. They're completely inactive. But you can switch them back on again. You can recover a functional T cell response to this epitope. By functional, I mean interferon gamma secreting. In various ways, by depleting regulatory T cells, which is this experiment, or by treating mice with checkpoint blockade inhibitors. But the point I wanted to make is that the therapeutic efficacy of all of these correlates very nicely with recovery of this specific response. In fact, we think that it's only this response that causes the tumour to shrink. We can predict the effect on antigen presentation as tumours begin to lose expression of tapacin. That's a very important phenomenon in oncology. You can stratify patients into sort of response groups or prognostic groups based on the expression of tapacin in their tumours. And what we'd like to know, of course, is which responses, which peptides persist as tumours begin to lose the expression of tapacin and MHC class I. And we can also predict the likelihood of presentation of peptides if we superexpress certain antigens inside cytosol. So in a kind of a vaccine setting. So what happens when you overexpress low affinity peptides, for example? And I want to finish just with this observation, which is what happens to the relative abundance of our two peptides, our high and low affinity peptide, as we simply express both to higher levels. We don't change the ratio. The ratio here is still 1:1 as we go up this diagonal. What happens to the relative expression at the cell surface? And what we find is that as the total amount of competing antigen increases up this diagonal, the output ratio changes. So you're more likely to present a low affinity peptide at very high abundance than at high level, than a high affinity peptide. And that again is regulated by expression of interferon gamma. So I pull those three examples together to illustrate why I think it's important to have a mechanistic model of peptide prediction, as well as our correlative models that we're all very familiar with. So I'm going to leave it there. Obviously, what we are working on is to extend the basic model here to include new molecules that we've discovered are important in this process, ERAT1 and TAP-BPR, for example. And then plug this model into other modules that we're working on with Microsoft and others to predict the likelihood of a peptide coming from a genome, a T-cell recognition module, the likelihood of a T-cell response given an abundance of peptide, and more sort of systemic modules, so the migration, the effective antigen-presented APC cell migration through the body, and the likelihood of T-cell activation given the integrated signal from positive and negative activated signals in T-cells. So thank you very much. This is the team who contributed to most of that work, particularly Rachel, who did all the quantitative pulse chase experiments. And these are not easy experiments to do. They're boring. You've got to do a lot of it in the cold room, and there aren't many people that can keep up with that. Denise, who did all the experiments with co-expression of different peptides to regulate their abundance. Gesser, who's working on the CD26 model, and of course our main collaborators, Andrew and Neil from Microsoft and Jörn from the structural biology department. Thank you very much.